2,950 meters below the surface of the Pacific Ocean. 300 atmospheres of pressure, the equivalent of 50 jumbo jets stacked on a single human palm. Temperature, 2 degrees Celsius. Permanent darkness, 0 photosynthesis. And yet, a colony of living vertebrates, not bacteria, not primitive microbes, fish, moving, feeding, thriving, stay until the end. Because what they found inside the cells of these creatures will rewrite everything science thought it knew about the limits of life. In 2011, a remotely operated vehicle began its descent into the Mariana Trench. No manned vessel, no submarine. A machine, because no pressurized hull built for human occupants could survive what waited at the bottom. The cameras rolled. The ROV passed 1,000 meters, 2,000. The water above became a ceiling of absolute black. The water below, the same. At 2,950 meters, the external pressure reached 300 atmospheres. To grasp what that number means, picture a single square inch of skin. Now press 300 kilograms of force onto it. Now multiply that across every centimeter of a living body, every cell membrane, every organ wall, every strand of connective tissue. Military-grade steel hulls deform at these depths. Welds crack. Structural integrity fails, not with an explosion, but with a slow, silent collapse inward. The water temperature registered approximately 2 degrees Celsius, just above freezing. At that temperature, most known biochemical reactions do not merely slow. They stop. Enzymes lose their functional shape. Metabolic pathways that sustain warm-blooded life simply cease to operate. By every standard model in marine biology, the place the ROV entered that day should have been sterile. A cold, pressurized void. The cameras disagreed. What they captured, pale, translucent shapes drifting through the beam of the ROV's lights, was not debris, not an imaging artifact. It was life, organized, vertebrate, unmistakably alive. The species recorded that day carries the name Pseudoleparus swirae, a member of the snailfish family, and the deepest fish ever documented on video. It does not look like a fish in any conventional sense. The body is semi-transparent, almost gelatinous. There is no rigid skeletal structure of the kind seen in surface-dwelling vertebrates. Bones are present, but loosely mineralized, pliable, soft. And that softness is not a weakness. It is the mechanism of survival. A rigid skeleton at 300 atmospheres would shatter under compressive force. The snailfish has no rigid skeleton to shatter. The pressure moves through it rather than against it. The animal does not resist the ocean. It accommodates it. But the external anatomy is only the beginning. The deeper answer lies inside the cells. The cell membranes of Pseudoleparus swirae contain an unusually high concentration of polyunsaturated fatty acids, lipids that remain fluid under extreme pressure. Place a human cell at equivalent depth, and its membrane lipids would crystallize. The membrane would stiffen. Permeability would collapse. The cell would die within seconds. The biological equivalent of a protein cooked in boiling water, denatured, permanently non-functional. The snailfish's membranes do not crystallize. They stay pliable. They keep exchanging materials. They keep living. Jasenyi. Then there is the matter of light, or rather, its complete absence. At 2,950 meters, no photon from the sun has ever arrived. The food chain that sustains the surface ocean, photosynthesis producing organic carbon, herbivores consuming it, predators consuming them, does not exist down here. The snailfish operates inside a chemosynthetic chain. Bacteria convert chemical energy from inorganic compounds into organic matter. The snailfish sits at the top of a food web that has never seen sunlight. In the most literal sense, it is an organism from a different version of Earth. And then there is the molecule that makes all of this possible. TMAO, trimethylamine N-oxide, is a small organic compound found in many marine species. In the snailfish, its concentration is higher than in any other known vertebrate. Tissue samples revealed levels that exceeded anything previously recorded across the entire vertebrate kingdom. TMAO functions as a piezolyte, a pressure counteracting stabilizer. Under the crushing force of the deep ocean, proteins tend to misfold. They compress, distort, and lose the precise three-dimensional shape required for biological function. T-MAO holds them in place. It chemically stabilizes the protein structure, preserving the molecular architecture even as hundreds of atmospheres of force press inward from every direction. Without TMAO at these concentrations, the snailfish does not survive. With it, the proteins fold correctly. The enzymes catalyze reactions. The organism continues to exist. This is where the story turns. TMAO saves the snailfish. That much is established. But TMAO, at the concentrations required for deep-sea survival, is not biologically neutral. At lower pressures, at the surface in shallow water, those same concentrations become physiologically disruptive. The molecule that holds proteins together under crushing depth begins to interfere with normal cellular chemistry the moment that depth disappears. The snailfish cannot survive above approximately 6,000 meters, not because the pressure is insufficient, because its own chemistry becomes incompatible with survival in conditions any other vertebrate would consider moderate. The molecular adaptation that lets it exist in the deepest places on Earth makes it toxic to itself everywhere else. The pressure that would flatten steel is not the snailfish's enemy. It is the snailfish's home, its requirement. The single condition under which its internal chemistry remains stable and functional. Remove that pressure, and the organism, not the environment, becomes the threat. There is one more detail buried in the genetic analysis. The genome of Pseudoleperis swirei carries an unusually high number of active DNA repair genes. The molecular damage occurring at these depths, from pressure, from cold, from the chemical environment, is continuous. The organism does not prevent damage. It repairs it, constantly, at a rate no surface-dwelling vertebrate requires. The snailfish does not endure the abyss. It runs perpetual maintenance on itself, every second, just to stay intact within it. 4 years. That is how long passed between the initial discovery of the TMAO stabilization mechanism in deep-sea snailfish and the appearance of any substantive public reporting on its broader implications. Several research teams filed inquiries with patent offices regarding applications of the TMAO-based piezolyte principle, pharmaceutical preservation, cryoconservation, the long-term storage of biological tissue at extreme conditions. The science existed. The potential applications were identified. The public record stayed quiet. For decades, the official model used by marine biologists set the maximum survivable depth for vertebrate life at 8,200 meters. The model was built on pressure calculations, biochemical thresholds, and the known limits of protein stability. It was presented not as an estimate, but as a ceiling. Pseudoleperis swirei has been documented at 8,336 meters. Not 8,200. Not at the ceiling. Beyond it. Which means the ceiling was never a ceiling. It was a number generated by models built on incomplete data, and the organism did not receive the memo. And here's the fact that carries the most weight. Every piece of cellular chemistry known about this animal, the TMAO concentrations, the membrane lipid composition, the DNA repair activity, was obtained from dead specimens, not from animals raised in controlled environments, not from tissue sampled under pressure. Every live snailfish retrieved from depth died on the way up. The decompression, pressure falling from 300 atmospheres to 1, destroyed them. What arrives at the surface is not a snailfish. It is a record. A collapsed, chemically disrupted record of something that was alive in a place where current biology insists nothing like it should exist. The official lower boundary for vertebrate life has been drawn, erased, and redrawn. At this moment, it holds no credible authority. At 8,336 meters, the line was crossed. At whatever depth the next ROV reaches, it may be crossed again. The next camera lowered to 9,000 meters may encounter something the TMAO model cannot explain. Something operating on a biochemical logic that has not yet been described. Something that refuses to fit the framework. Because the framework, as the snailfish demonstrated, was built on the assumption that limits exist. No rules have been confirmed. No floor has been found. And everything currently known about the deepest vertebrate on Earth was learned from its corpse. If this changed the way you see the limits of life, subscribe. The next descent is already being planned. And whatever the cameras capture down there, you will see it here first.